Agarwal, who is an associate professor of biomedical engineering at the University of Miami, so just down the road. He joined the University of Miami as an assistant professor in 2014, and prior to coming to UM, he was a postdoc at the Wyss Institute at Harvard, just downstairs from me, so we probably overlapped for just a bit, maybe a year. Then before that at Columbia University. He completed his PhD at the University of Florida in material science engineering. His research focuses on development of platforms that mimic in vivo conditions that allow him to do mechanistic studies of different diseases, tissue physiology, pathology, drug screening, stem cell differentiation, and general mechanistic studies. This work has been supported by the NIH, the Wallace H. Coulter Foundation, which a lot of us know, and Mallinckrodt Pharmaceuticals. So happy to have Dr. Agarwal here with us today.
Thank you. My postdoc advisor used to say, most of the opportunities come to you if you show up, if you know where to park, and if you're cheap. And that's what happened today. I knew where to park. I showed up and I'm very cheap. So I have an appointment in biomedical engineering and most of BME labs at the University of Miami. And I would welcome any of you all if you're interested to visit. I can arrange a tour. Are at the medical school. And so that really embedding physically the lab at the medical school brings a level of relevance and also urgency to the research we do and also resources. So I have appointments at the Cancer Center and the Diabetes Research Institute over at the medical school. We also got, less than a year ago, a $20 million naming gift to a urology department, and that created a urology institute. And the mission of this institute and the way the dollars would be spent would be in engineering research around urology. And there's a lot of stuff to do in urology, so if anyone's looking for robotics, image analysis, postdocs, and things to develop tools for surgeries, please come talk to me. I'm the director for the engineering side for urology. So for my research lab, the problem that I want to submit to you is of drug development. People have heard of organs and chips? A little bit. The idea is if you take a look at drug development, a pharma company might typically spend 15 years and $2 billion before it makes any money on any drug candidate, and the bulk of that money and the bulk of that time is spent in preclinical testing, which means before human testing. So everything they want to bring to human testing, they want to make sure it passes and doesn't fail. All the failure would be better to fail early because it would be much cheaper to fail early. So how do you fail early? Actually, let's talk about this graph as well. On the y-axis are the number of drugs you could bring to market if you're a company with $1 billion investment, adjusted to inflation. On the x-axis is time on a linear scale. So the y-axis is inflation-adjusted number of drugs per $1 billion investment on a log scale. So it's pretty shocking, right? The exponential decline in return on investment, and we're right, so in about 2000 we were at one drug per $1 billion investment. So we're well into like more $1 million. So it's not cheap, and that gave rise to the mothership that I come from, which is the Wyss Institute, where we had a $35 million DARPA contract to develop 10 different organ chips and connect them all together, have them talk to each other for 4 weeks and do drug testing. And the paper that spurred the imagination for both DARPA and now follow-on funding agencies was a lung on a chip, which I'm sure many of you have seen these movies, where there are simple monolayers, stacked monolayers, epithelial, endothelial monolayers to create the barrier function of the alveolus, and there is this biomechanical stretch that makes the lung breathe, really. But all it does is coax the cells into believing that they're still part of the alveolus, and that leads to better recapitulation of their responses, especially to drugs or challenges to toxins, for example. Now, when I did this as my postdoc, I was, so this was all the postdoc work from my postdoc labs. And when I was starting my lab at Miami, I had this challenge of differentiating myself from this, from this juggernaut of organs and chips. And the way I thought I could differentiate my research lab in Miami was to not focus on these very low volume microfluidics, where it's good for culturing monolayers or stacks of monolayers, but to go away from photolithography-based fabrication of these low volume microfluidics to large volume microfluidics for doing organoids, spheroids, pancreatic islets, tissue samples like ex vivo slices of tissues. And not have to use a clean room, and not have to use this material, which I'm sure many of us are aware is called PDMS, right? So we love to hate on PDMS because it's quite easy to use, it's transparent, it's bioinert, it's cheap, but it's absorber of many things, especially hydrophobic molecules. And most of your drug candidates are small hydrophobic molecules. So if you're using PDMS to do organs and to do drug testing with organs and chips, the housing of the material will absorb most of the drugs you put in. So I set up my lab here in 2014, and the first paper out the gate was the philosophy of creating microfluidic devices from acrylic, from polystyrene, the inert plastics that we culture ourselves on. And we would simply create features into acrylic by conventional machining techniques, like CNC micromilling machines or laser cutting systems, that would create these housings which were 2 millimeters tall, 2,000 microns of clearance, which would be enough then to hold, let's say 1,000 micron organoid or a cancer spheroid or a pancreatic islet. And it was this ecosystem of 3D organoids that conventional organs and chips was not addressing. There is no way for you to manually place these large volume large diameter organoids in conventional microfluidics and then have them be perfused. Now, the problem with raising the roof on a microfluidic device then is you lose control over laminar flow. You cannot ensure laminar flow if you have high clearance, right? Now, so the way around that was to build in geometrical features like a chamfer cut at the entrance or an obstacle at the ceiling to really make sure that anything that you're flowing in actually hits the area where biology is, and then narrowing out the exit, for example, so that everything that the biology is producing, let's say secretomes or insulin or whatever, gets collected quite rapidly from the outflow, and there's no dead zones. And so we solve for the flow dynamics within the chip. We couple those flow dynamics with the production of, let's say, insulin by pancreatic islets. And through the combination of these two, we come up with idealized geometric prediction for the device geometry, and then we take that 2D prediction, convert that to 3D CAD in SolidWorks, and go out and fabricate it using the suite of micromachining tools in the lab. And then you end up with 2 pieces of plastic that snap into each other like Lego pieces. These 2 pieces of plastic are separated by custom-made green gaskets. So that's in green over here. And these gaskets are like O-rings, right? They're these compressible rubber O-rings that will compress under the clamp, under the mechanical force of the clamp that we also get micromachined by a company in the Netherlands. So now you've got plastic-based microfluidics that you can reversibly place your biology in manually. When it's ready for perfusion, close it, clamp the clamp, and now you're ready for perfusion. And so it's quite simple in idea in the conception, right? So you've got 3 wells, 3 channels, 3 inlets, 3 outlets that all get connected through these wide ferrules in the clamp. So with this, the first funding was for type 1 diabetes. This was in collaboration with Dr. Sherry Stabler, who's now at UF, who recruited me to Miami and moved as soon as we got our first grant to UF. So it's a multi-PI UO1 to do type 1 diabetes on a chip. So I learned from her how to, get pancreatic islets from rodents, and we also get pancreatic islets from human donors. And the typical experiment that you want to do with pancreatic islets is to starve them and then give them breakfast. And then on the y-axis, measure how much insulin those islets are producing in response to breakfast. And then you should see a big spike in insulin, so that's your, you feel satisfied, you stop eating. But even after you stop eating, you want to have the insulin be maintained at a plateau. So you don't want that insulin to be at a super high level. So you want to see a quick spike, but under high glucose, you also want to see a plateau, which is not at 0, but at a non-zero value. And only when you start starving them again, do you see the insulin go down. So that's a healthy glucose-stimulated insulin response. And the problem that we were trying to address, and that the NIDDK asked us to address, was these islets are meant for transplantation in the bodies of type 1 diabetics. Type 1 diabetics have an autoimmune attack, they don't have islets anymore, and there's a clinical islet transplantation program at the University of Miami, for example, and at other centers. But the challenge is you have to line up a a recipient for these islets pretty soon after you get a source of human islets. So let's say there's a car accident, right? There's a source of pancreatic islets. Now you have to line up a patient, a type 1 diabetic patient, who will receive these islets within almost a day, because these islets are declining in function rapidly. One of the reasons these islets are that the shelf life of these islets is so short is because in the body, the islets are highly vascularized, but the process of obtaining of isolating them kills all that internal vasculature, and they become oxygen deficient and they die. So the decline in function is a big, major issue, and the grant Sherry and I, Dr. Stabler and I have, is to improve the shelf life of these islets. So again, as you see, within the same day, within my microfluidic device for human islets, we could very clearly show the decline in insulin production within a week. So the next version of devices was published last 2 years ago now in Science Advances, where the first version of device was the solid plastic device, but instead of a solid plastic device, we made a through cut on the floor, and at the place of the through cut, we stuck an oxygen permeable membrane, the properties of which were bioinert, and we understood the oxygen transmission, like diffusion properties. We solved for those oxygen diffusion properties first. We stuck that highly transparent thin membrane on the bottom, and then we measured oxygen through embedding fiber optic oxygen spot sensors so that we could verify that our predicted oxygen permeation at the site of islets was enough for ameliorating the hypoxia that islets suffer from under high glucose. So with this setup, Sherry's lab and my lab. So one of the other things is my engineering devices are meant for consumption in my lab, but we also make sure that others can use them. So the big thing, like we have it in bold letters in the lab, make your devices simple enough that even a biologist can use it. Sometimes biomedical engineers end up making like these complex devices and they don't get adopted, and ease of use is one of the big things. So Sherry and I, for example, are able to manually load both naked islets and islets that she encapsulates in some biomaterials that protect against an immune attack. And this manual loading and casting of islets in a microfluidic device is a huge advancement, because you don't want to flow in the islets and expose them to shield. Then you clamp the device, you have the device running in an incubator. Every morning you bring the device out, you challenge it with glucose, high glucose, low glucose, or you just put that on a microscope, and then you put the clamp back in the incubator. And so here again are very stark results from a chip that's not oxygen permeable, which is fully plastic. Unsurprisingly, there's a lot of death, and an oxygen permeable chip where you see still restoration in longevity. You also see it in the function, quite stark differences between an oxygen permeable chip and a plastic chip. The real proof is with human islets. And again, with human islets, we were able to show increase in functional lifetime of these islets to a few days, and now we're up to 2 weeks after this paper got published within a very simple microfluidic device. And so the next version, so that was the first 5 years, that took 5 years. And so the next version of this U01, which we luckily renewed, is to now really do the islet immune. So really now take this validated platform and bring it to town for lab applications. So what are the questions we're trying to answer? Dr. Stabler's lab is one of the leaders in assessing different biomaterials that could protect the islets against an immune attack. So she's doing a lot of biomaterial assessment for encapsulation of islets. In collaboration with Harvard Stem Cell Institute, Dr. Doug Melton, who's one of the creators of embryonic stem cell derived islets. Have you guys heard that story where he sold his company Sema Therapeutics to Vertex for $1 billion cash? So Vertex is bringing those stem cell derived islets to clinical trials. There's active phase 1 and 2 clinical trials going on with that technology. So along with him, we are testing the stem cell derived beta cells within these microfluidic devices to see if there is an insulin production over time. And the very exciting part also is with added collaborators who are immunologists, beta cell biologists, and stem cell biologists. So Clay Matthews and Todd Briscoe, both at University of Florida. We are modeling an islet immune attack on the chip. So in this movie, which is going to loop over time, on this top left, you will see immune cells sourced from the blood of the same patient that we derived these beta cell clusters from, come in, home towards the islet, and destroy the islet. here, you see this rapid islet immune attack in a patient-specific. So both these cells are derived from the same patient, right? And that opens up opportunities for us to now test, right? What kind of antigen activation, what kind of protection methods, what kind of anti-immune cocktails does each patient need to be on?
Can I ask a question?
Yes, sir.
How do you know you haven't introduced any new antigens onto the islets that,
100% you're right. You're right. You're absolutely right. We're doing this now with not patient-derived cells, but like actual pure lines that from the same source we would derive.
because you do so much manipulation that
Yes.
sorry.
Yeah, so this is like literally a movie from not too long ago. So we just finished this. And that's also one of the reasons I point those questions to immunologists a lot, because there's only so much we understand about immunology, and they are very like deep in
Well, diabetes is an immune disease.
it isn't. Okay, so from the lab side, the devices have found interest from a lot of academic collaborators. So this design, build, test, rapid design, build, test without using a clean room is useful for us because we can iterate within half a day between different designs. I've partnered with companies in the Netherlands who make the clamp, and then a fluid handling company in France called Fluigent that makes, they call it the Miami box, and they sell this Miami box for anyone who wants to use our devices for the regimes of glucose-stimulated insulin response, all embedded into one system. Again, to make it easy enough that a biologist can use it. And we spun out our own company, BioVitro, and through BioVitro, we were able to service collaborations at Bandy School of Medicine, WashU St. Louis, Hebrew University, Jerusalem, UPMC, and Indiana School of Medicine. Our chips are living in their diabetes research institutes, and they are using our chips and actually publishing with our chips. So one of the first papers that came out, and there have been follow-up papers, was from the Diabetes Institute. We are co-authors on it because we didn't sell them the chips. The company was still new, so we were still an academic collaborator. Al Powers and Marcela Brisova were able to transfect alpha cells, beta cells, delta cells that make up the pancreatic islet differently with different ion channels, and stimulate different ion channels with different wavelengths of light within our device, and collect the corresponding hormones. So alpha cells, beta cells, delta cells would produce a different hormone, right? That co-regulate each other. And just the simple function of being able to manually place these, they call them pseudo-islets, human islets in a chip, in an optically transparent chip, and have perfusion to collect secretome. It's such a simple idea for us, right? But it's a game changer for us. So I was very lucky that they got this nice paper out using that chip. Okay, so that's the funded study number one. I could pause here and take questions, and then move to the next one.
I do have a question. So the size of these organoids is what?
It varies. That's a problem. So rodent islets could be 150 to 200 microns. Human islets could go up to a million.
Because if you get more than 300 or 400, then if you don't have a vasculature, you're not going to get, oxygenation. I mean,
Yeah.
a couple hundred, you probably get plenty of diffusion. These cells, are they very active in terms of their metabolism? So
Especially-
uses a lot of
Yes.
oxygen and.
So one of the reasons why we had to build that floor for independent oxygenation from water
Yeah.
was we couldn't oxygenate the media itself fast
Oh,
enough.
sure, but then there's a limit to the size in which even that additional oxygenation on the floor isn't going to.
I agree,
matter.
and there
You know,
is a lot
because
of-
I'm coming from a cancer side where, cancer organoids where you always have this hypoxic
They do too.
center. Yeah.
So there's a lot of death in the center.
I see.
And it's the process of isolation from the pancreas that causes death. of
That's
the
part of it.
vasculature.
It's just
Yeah, there's no way around it. I think the people who are working towards implanting islets in human patients, that's the fight I'm not fighting. They are thinking through strategies of pre-vascularizing
Right,
these
yeah.
constructs and then transplanting them, and hoping that the pre-vascularization anastomosis with the host's vasculature, and then there's a nutrient exchange. The vasculature is also important for bringing glucose to the center of the islet and bringing insulin out. Yeah, that's right. So the second major funded effort, again, this is like my academic ADHD, is a completely different effort. It's funded by NCI, also U1, also multi-institutional, between WashU, Caltech, and UM. I'm the contact PI on this one, where we are tackling the problem of circulating tumor cells. So this, again, is a crowded field, but the idea here is Every cancer patient, depending on the stage of cancer, would put out cancer cells in the bloodstream, and those cancer cells, because they're in circulation, are called circulating tumor cells. These circulating tumor cells, most of them die or do nothing. A very small fraction of them choose to extravasate out to a distal organ. Most of the extravasated cells are senescent and die and do nothing, but a small fraction of those extravasated cells can flip and start causing micrometastases and metastases in distal sites. Especially for breast cancer, there is a lot of good clinical advances in handling primary tumors. So 90% of breast cancer deaths happen because of metastasis. So it's this metastatic disease that we're trying to replicate on our chips. Through a technology that I did not develop, we are able to isolate all different cell types that should not be in blood, that we do find in blood. So obviously circulating tumor cells, we find cancer stem cells, we find circulating cancer-associated fibroblasts, and even NDSCs. And it's the combination of these cell types in the blood that's been shown by work before me to be a poor prognosticator for survival. So again, through technologies that I didn't develop, but my med school, again, the chair of pathology who recruited me to Miami has now moved to Washington, but we have a grant together. It's the story of my life. He developed these very simple microfilters, and these are size-based exclusion microfilters where cancer cells, the size of the filter is sized so that white blood cells and cancer cells get stuck if you push blood through it, and the red blood cells go through. And there's been a family of iterations he's developed. What I helped to do was make this assay and make it standardized. Take this assay and make it standardized. So we spun out a company that where you can buy these microfilters, run fresh blood through this cartridge, through this holder, through this microfilter slide, and then when the blood is pushed through this, you can capture cancer cells onto the microfilter slide, which you can stain and image, and that's how images look. It's a pure size-based exclusion, so it's cancer agnostic, it's antigen agnostic. And so we've done, his lab and my lab have now done studies in both solid tumors and non-solid tumors. And it's quite remarkable when you actually can see different cell types from the blood of human patients. And for the context of my grant, we are working with breast oncologists, surgical oncologists, where we're enrolling stage 1, 2, and 3 breast cancer patients who every 6 months give us a blood sample. Actually 5 tubes per draw every 6 months. 2 tubes are meant for circulating tumor cells and associated cells. 2 tubes are meant for cell-free DNA, so this is the cell-free circulating DNA. And 1 tube is meant for extracellular vesicles like exosomes. And it's the combination of these markers we're hoping would produce a better prognostic index for management, for fate of these cancer patients. So we're actively recruiting both at Miami. We were only doing Miami, and then my other PI moved to WashU, so now we're also recruiting at WashU.
I guess your success scares them off, and they have to set up shop somewhere else.
I don't know. This must be something about me. Maybe it's the opposite. Something scares them off. But it's good because the grant actually works better, because now we can validate our platform in 2 sites. One of the other papers we published early on and a patent was to release these circulating tumor cells from the filter using a coating which I'm sure everyone knows. It's a specific polymer, PIPAM, which is temperature responsive. It loves water at room temperature and it hates water at 37°C. So all we do is pre-coat the filter with this coating and release the cells at 37°C. So now we've got patient CTCs to play with. That's a game changer. And so for every vial of blood, we can now recover circulating tumor cells. And in my early naive days, I used to say, well, if I have the cells directly from the patient without cutting into the patient, and if I can grow those cells, I can do chemosensitivity assays for each patient, right? And this is the promise of liquid biopsy. You're not biopsying by cutting someone, you're biopsying someone just by collecting a vial of blood. CTCs is really very hard to grow, and CTCs also, are interesting but very high maintenance. So one of the things my previous graduate student who has graduated now, she thought of creating a bone marrow niche from the same patient to get the circulating tumor cells from. And her bet was that it would be something to do like by introducing a metastatic site and with the signaling molecules, and of course she had a favorite molecule, CD146, that has been shown to be the mediator of literally pulling these circulating tumor cells out of circulation and bringing them to a bone site through the mediation. The mediation is through these perivascular cells or pericytes. So we would get mesenchymal stem cells, bone marrow-derived mesenchymal stem cells from the same patient as we do CTCs. From those MSCs, we would create bone. From those MSCs, we would create pericytes. And the idea was to just like do this in a chip. And she ended up with this crazy looking chip, which is super impractical. So we're trying to simplify it. But as I said, we have a steady supply of patient CTCs. And she created a channel on the bottom where circulating tumor cells would be in recirculation in the yellow channel. At some point, the circulating tumor cells would notice that there is a bone marrow environment of the same patient living somewhere on that chip. That bone marrow would be separated from the recirculating CTCs through a vascular membrane, right? So we got endothelial cells and pericytes across a transfer membrane that we built into the chip. And on the other side, not in physical contact, but in liquid contact, is the bone that's enriching the liquid milieu of the circulating tumor cells. And hopefully at some point, CTCs will go up and cause metastasis to the bone. We spent 3 years, it's not happening, but we are seeing some very interesting, we published this a couple of years ago, we have seen very interesting surface marker-based antigenic shift of cancer cells when they're put in conjunction and sequentially, first in conjunction with pericytes, and then in conjunction with pericytes and bone. And you know, the cancer cells go through this phenotypic shift when they're just in the chip where there's no bone, and then when they're in the chip when there is a bone. And we're still trying to parse out the overexpression of markers that they're robustly showing linked to, basically a mesenchymal to an epithelial phenotype. And so this grant is in its 4th year now. Hopefully we'll be able to renew it. And in the renewal, we have submitted the idea of like a robust chip, hopefully that lets us dive deep into disease mechanisms, over time create CTC biospecimen from each patient, and then do chemosensitivity assays for that patient. So those are the 2 funded stories. I have a lot of unfunded work that's still brewing, so I can wait here. Yes?
I have a question
Yes.
for the identification of the prognostic
Yes.
biomarkers. Do you control for the kind of therapies that these patients are receiving?
Yeah. So they are in 2 arms. So there's an adjuvant arm and a neoadjuvant arm. Sure. Good question. Okay. So maybe I can skip this paper and show you work which we haven't published yet. Because it's a PhD research and you're doing posters, the point is to present work that's not ready yet, right, for prime time. So this is the sausage part of the sausage. We got interested, again, playing to the microenvironmental strengths of South Florida. South Florida is one of the areas where hepatitis, like infectious diseases are, still there. HIV is still there. Hep C, even though we have good drugs for hepatitis C, is still there. And these are needle users from our parents' generations who are carrying Hep C, they don't know they have Hep C, and hepatitis C over time progresses to liver cancer, hepatocellular carcinoma. And that's how they present to the clinic, and then they get surprised they actually have Hep C. They've had Hep C all their lives. And so we had some internal money to do host response analysis in virology, and to mimic the progression of a Hep C infection to liver cirrhosis, and then over time, liver cancer. Again, there are very good drugs now for Hep C, so this should resolve the issue, but it's not in South Florida. So my graduate student who just graduated and got a job in industry, he developed a liver chip where he again, from the context of organs and chips, liver is the organ that makes most sense because everything is hepatotoxic. So there have been a lot of liver chips that have been developed in the past. To enter a crowded field like this, you have to be very careful and see what is your secret sauce that no one else has. So he wanted to create a simple to use chip where hepatocytes would sit on the top and not get exposed to shear, because typically hepatocytes, if you take a look at the cross section, produce bile that gets, that's not flown out by active shear, active flow processes. So his hepatocytes live on the top of a membrane, and on the bottom of the membrane we flow in liver. We culture liver specific endothelial cells, and over time we build in the the resident immune cells and stellate cells. We do it, like every chip developer, we first try and do it with easy, cheap cells. So the cheap but stupid cells are HepRGs, immortalized cell lines, and HUVECs, immortalized cell lines. Not very replicative, but when you're trying to perfect the chip, that's what you use. And over time, you want to obviously move to primary human hepatocytes and liver sinusoidal endothelial cells. So I'll show you results, very quick results with both of them. His chip is quite simple. It looks like a multi-well plate at the exact locations of the actual wells of a normal multi-well plate. The middle row is for the culture, so it's got 8 culture wells, and on either side of the culture well are the channels that are the inlet and the outlet for the lower endothelial barrier. So he's got an endothelial barrier running on the bottom surface, and then he's got a membrane on top of which are liver cells, and he has it repeated 8 times over. So again, we solve for the fluid dynamics and like the flow of regime, the flow of the. So he controls. Excuse me, this is important to show. So the pressure difference between the left and the right will drive the flow from left to right, for example. But then at this interface, the liquid has the decision to make to go to the outlet or to go up towards the hepatocyte. Or the hepatocyte media has a decision to make to go down. And it's the difference in the pressure head at this interface that would derive whether the hepatocytes are talking to endothelial cells or the endothelial cells are talking to the hepatocytes. And so through this clever but easy to use strategy, he was able to culture primary human hepatocytes in the chip. We were able to show, and this was, I was very surprised, I made him repeat this like at least 10 times because I was shocked at how well it works, so I was obviously very skeptical. Primary, hepatocytes are notoriously hard to grow. They're also high maintenance cells. He created 4 conditions: a normal monolayer hepatocyte culture that all hepatocyte biology labs do. So this is the state of the art. He also mixed in hepatocytes with endothelial cells as a co-culture, as a control. He then made the chip but did not perfuse the chip, that's the blue. And then he had the actual chip with perfusion. And for different markers that would show you the function of hepatocytes, which is the enzymes they produce, over 1 week, 2 weeks, and even 3 weeks, the chip worked well and better. And so this is in review at a good journal. Hopefully they'll accept this. We've shown it's capable of imaging. We've shown that it also increases drug sensitivity. If you're building a hepatocyte, you better show that the hepatocyte metabolizes drugs and is sensitive to drugs. So we've shown that in the perfusion chip configuration, the hepatocytes are more responsive, like they produce more enzymes, so we knew they would metabolize drugs better. And so in response to drugs also, we show CYP3 activity. We also put, so those were data from non-human hepatocytes. We also put primary human hepatocytes. Again, these are, islets don't divide, hepatocytes don't divide, so we're working with very hard to maintain cells that don't have good shelf life. And so for primary human hepatocytes, we had mixed results, but we did show extension of life and function. We went a step further and infected them with Hep C. And with Hep C, you want to make sure that they're getting infected through the right pathways. So ISGylation is one of the pathways through which hepatocytes take up Hep C and respond to Hep C. And so we showed first that hepatocytes in our chip do take up the virus, and then actually respond to antiviral therapy in a responsive manner through the expression of certain markers. So a lot of data in one manuscript, where we established first a good culture method, that it works for human hepatocytes, that they take up the virus, and they initiate the right immune pathways. And then they also don't upregulate the off-target genes that they should not be doing. So a lot of housekeeping genes as well. So that was my liver story, and we're hoping now to take this further and apply to NCI for creating hepatocellular carcinoma. This is the grant that did well. We got up to a 12th percentile at NCI. Only PIs know that, or PIs would understand this. And the Cancer Center was very supportive. A 12th percentile is still far away from NCI funding line, and they gave us the money to keep doing the work. We did more work, and now it got not discussed. So that's what happens with NCI. Anyway, so that's my one of the unfunded stories. This is the story that's closest to my heart, where again, this was led by a PhD student who's now graduated, Rachel. She started talking to a neurologist where he runs a clinic for a rare genetic disorder that manifests as a neuromuscular disease. The name of the disease is Charcot-Marie-Tooth disease, CMT. Not many people have heard of it. It presents clinically like ALS. No one understands whether it's a disease of the neuron, disease of the axon, disease of the junction, or disease of the muscle. And so from iPSC source neurospheres that the neurologist gives us, we created a chip that could isolate the axons of the neuron from the endings, and then have the endings connect with skeletal cells of the same patient. And hopefully then we could stimulate the neuron and ask the question, how is the transmission? Or ask the question, how is the connection? Or then ask the question, how is the twitching? Right? So all the stages of a neuromuscular junction. To do this, she first had to discover a biomaterial on which, again, these two high maintenance cells could be happy. And like this was a mixed project. This was a project of mixing different biomaterials and coming up with a hybrid biomaterial, which was published in Biomaterial Science. She also had to discover the media in which these two cell types would be happy. That's also not obvious when you try to couple tissue 1 with tissue 2. And then once she had a biomaterial and then she had a media, then she actually started building neuromuscular junctions. We recorded through microelectrode array activities from both neurons and muscle. Then we put them together. It worked quite well, right? So you've got a neurosphere, it's extending axons, and it's connecting to skeletal muscle. You take a closer look, and you see a deep innervation of skeletal cells. You see a nerve ending, right? And forming in red is the staining for acetylcholine receptors. Acetylcholine receptors are skeletal cells assembling these receptors in response to having a nerve ending close by. This was very exciting for us. Even though the staining looks like crap, but like this is actually a really good result. And then, in addition, and co-localized to acetylcholine receptors, we could also stain for synaptic resonance. So again, as you can tell, this manuscript is in review, but this is the result that we were most happy about. So here she is measuring acetylcholine cluster size that the skeletal muscle is assembling. And when she constructs the chip from healthy donors versus donors who have this Charcot-Marie-Tooth disease, we see a quite robust, reduction in acetylcholine receptor cluster size. We don't know yet what it means for muscle function. We're trying to do the twitch experiments with muscle. My training is in cardiac muscle, so I'm trying to adapt the same techniques to skeletal muscle. So hopefully in the next iteration and after funding, we'll be able to actually show that a neuron not only doesn't talk to the muscle very well, but the muscle doesn't twitch in response to neuron stimulation. So I have one more story, Josh, should I keep going? Sure. So I'm just presenting vignettes without going into details. The last story is funded, actually just got funded. It's a retinoblastoma on a chip. This is the cancer that affects young kids with that in the eye, and if not caught early, it can be fatal. If caught early, they lose the eye. So it's like both outcomes are pretty bad. We know the mutation that causes retinoblastoma, especially in the developing world, the way these kids, kids can't tell you, right, that they have cancer, so the way the kids present is these kids would not have the red eye effect when their parents take the pictures of the flash. So especially in India, right, like the parents would come and say to the pediatrician that only one eye has the red eye effect, and the other eye doesn't have the red eye. And that's how, that's how it's gone. So the challenge became, how do we start growing human retinas in a lab? There's retinal tissue that comes up for research purposes, but not very much, and it's not the whole retina. So my collaborators at the Bascom Palmer Eye Institute, another center of excellence at the medical center, and I started looking at how the retina develops over time, and it's very interesting. The front of the retina experiences 21% oxygen, right? The back of the retina experiences almost 0% oxygen. And so we wanted to stably create this oxygen gradient that's been implicated to drive the differentiation and self-assembly of all of these complex cell types in the retina. But to create that oxygen gradient, we had to discover a lot of different, so we had access to human iPSC, actually human embryonic stem cells right now, but over time human iPSC derived retinal organoids. And what we wanted to do was, in a chip, create this gradient over time. But what does over time? So again, this work is led by Florida's #1 biking champion, apparently. So she won that #1 position a couple of months ago. Emma Drabe. She's from the Netherlands, so biking is in her genes. She created a chip with an oxygen gradient over 6 months. Apparently, the differentiation takes 6 months. So here is a chip. She has this large organoid. Again, the floor of the device we flush with 0% oxygen. The top of the device is open, and so she's got 21% oxygen. And over time, that establishes an oxygen gradient in Z, which is perfect for us. And so this is how it looks on a computer. Obviously, this is her design of the chip. She created a microscope slide format, but within that microscope slide format, she had 60 organoids differentiating for 6 months, where the floor of the device is flushed with nitrogen, pure nitrogen, 0% oxygen, and the roof of the device is at 21% oxygen. She solved the console. So the chip lives in an incubator. Because it's 6 months, you don't want to monkey around with it too much. You have all the controllers sitting outside for both the gas and liquid perfusion. And then she has her little babies growing for 6 months. She verified first through console that there is indeed a nice gradient, especially at the location of the retinal organoid. And she verified those predictions by actual measurements, by having the same oxygen spot sensors along Z. So along the length, along the height of the organoid, she does get a nice gradient. This is the protocol. I mean, more power to her, but it's like this is a really long protocol.
Especially for a graduate student.
Sorry?
Especially for a graduate student.
Yes.
It's excruciating.
It's excruciating. Yeah, so 4
I need
months.
experiments in 24 hours. I need results in 24 hours.
Yeah. 4 to 6 months is the protocol. Also, she does get these optical cups growing in her retinal organoids. We got very excited, and then we took a cross section, and we are seeing some early indicators that there is this nice organization of differentiated cells, self-organization of differentiated cells at different time points after the end of differentiation protocol. So very early days, she's starting to do RNA-seq at the single cell level to parse out what kind of cells are appearing at what locations. It's a lot of arts and crafts, but we're hoping that the chip is robust enough and actually causes a nicer organization. Over time, she will cause through CRISPR-Cas the RB1 mutation known to cause retinoblastoma. We're also playing around with the timing of inducing the RB1 mutation is interesting because retinoblastoma also is sporadic and hereditary. So if we cause the RB1 mutation on day 0, that would mimic the hereditary retinoblastoma. But in some forms of retinoblastoma, it's also shown that it's a sporadic introduction of RB1 mutation. Thankfully, this grant is funded, so it's actually not just my startup. So that's all the stories I have. I'm hoping you guys got excited about the potential of organs and chips and got a good overview. The real power of organs and chips is realized when you couple organs to each other. So the 10-year vision of the lab is to have multiple organs talking to each other, have the liver upstream, for example, so that the liver metabolizes the drugs and the metabolites then hit different organs. Just from the neuromuscular junction project, I can appreciate how difficult it is for doing simple things. Simple things like in our body, the blood perfuses all the organs and we don't think about it. But in our chips, we have to find a different media for each organ. From a pure mechanical engineering, more organs, the more valves and pumps and tubings you see, the more ways of failing through air bubbles, sterility losses, and leaks. From a scientific point of view, it's not clear how you scale each organ chip to each other because it's not clear how each organ is scaled to each other. We don't know how a brain and a heart and a liver and a kidney is scaled by nature. Is it by mass? Is it by volume? Is it by perfusion rate? Is it by metabolism? And also then, how do you extrapolate the response you get from the chip to the response you would get from a human? So this in vitro to in vivo extrapolation is a very scientifically rich area that hopefully keeps us busy and fun. From technology adoption point of view, I will say this is our enemy. The enemy are these super simple multi-well plates. Cells are sitting on these very artificial environments, stiff plastic, no perfusion, no electrical stimulation, no stretch, but they're very cheap and they're very available. right? So the cost and throughput is good. Our chips and most of the chips my colleagues are developing are very smart. So they would like to believe that they're very sophisticated, but if you take a look at the cost or the throughput, it's terrible. I mean, the throughput of my chips is terrible, except the retinoblastoma chip, it's an N of 3 or 1 from one chip. So I'm like being very purposeful through my startup company and conversations. What was the name of the program in which we had to talk to customers? ICOR. We got to participate in the ICOR program, and they all told us, you need to make it simpler and cheaper and more to be of value. So one of the things that came out of ICOR was to create this fluid handling robot that actually 4 senior design students from the department built, where there's a chip sitting here, and there are different sources, and through a fluidic manifold and a peristaltic pump, it switches between different sources. And when it switches between different sources, the output collection is also switched to a new collection board. So there's an XY Arduino motor for the collection plate, and there's a fluidic manifold for switching between different sources. So quite simple, but like the dimensions are such that it fits into a 96 well, into an incubator. But here, these students are switching from a yellow source, and then they switch to recirculation mode. So the yellow source is going back into the yellow reservoir, and then they switch from a yellow source to a green source and output it to these wells. So that was quite, and so this is that Miami box that French company Fluidic now sells commercially. These are the 2 companies. They should be on the top of the slide deck, not as an advertisement, but that's the cancer diagnostic company that's doing well. We get blood shipped to us overnight from different sites, and the organ chip company is still here. We're writing SBI analysis to get it funded. These are the students. I'd be amiss if I didn't mention the collaborators. My cancer people are Richard Cote, who's now at WashU. My liver person is Emmanuel Thomas. I didn't present my glomerulus and kidney work, but the head of nephrology, Alessia. My neurologist is Mario Sukota. My retinoblastoma person is Dan Perez. And we recently got a grant as a co-I, not as a PI, to do pancreatic slices. Not do islets, but like the whole slice of pancreas on a chip. And that's with Juan Dominguez Mandala. And that's who's keeping the lab from.
Questions, Dr. Agarwal?
That was so interesting. I have like a general question. Like, you're all over the place with the type
Yes.
of diseases you study. Like, how do these ideas just like?
No, it's the microenvironment.
Oh, it's the environment.
Right. It's the environment of who at the medical school has a compelling idea, has resources, and like the students then pick.
So it's more of like you have the device expertise. Right?
Yes.
And then
We will back engineer something to fulfill the needs of the experiment. Yeah. And there's more to that. More to it.
because there's a lot of background to understand all these different diseases, right?
Yeah. So every PhD student here is co-advised by a different medical school PhD. So, absolutely every student has a co-mentor. Yeah.
So how do these projects begin then? I mean, so where do those conversations start, and how do you develop those collaborations?
Just like you do, a lot of meetings, brainstorming. We invite people to a lab meeting all the time, and they present to the lab. And,
On the technical side, so especially with the neuromuscular coupling, are you measuring the skeletal muscle contractility?
we're starting to, yeah. So I didn't show that result, but they all have fiduciary markers, and through just motion analysis. So that's the dumb way to
You
do
measure
it.
with the cells there? Like,
Yes.
okay.
But the other way is they're all sitting on a multi-electrode array. So it's a 64-channel multi-electrode array where the neurons are sitting on the north, then the extend axons, and then the skeletal muscle. So we can see locationally which channels will be recording activity from the neurons and which channels will be recording activity. It's hard to isolate them right now.
The stiffness of the substrate you're sitting on doesn't provide.
It's the
cardiac
same
resistance.
substrate.
Okay.
It's the same. It's that gelatin laminin biomaterial. It's 7 kPa. It's too stiff for neurons, but it's quite perfect for skeletal muscle. So they love skeletal cells love gelatin, collagen, and neurons love laminin. So we have to like give them both. Can I advertise the program in the last 5 minutes? So I thought I could just talk about this. So, you know, after tenure and after summer 2020, where all the universities realized, right, they need to do more anti-racist things, and especially rich private colleges like University of Miami, which looked around and said they're not doing much in terms of diversity. They asked for grants to study anti-racism. We wrote a grant and they gave me $100,000 to fund Black undergraduates in different research labs. So this is a year in making. And in response, so I fund Black undergraduates to work in different labs at $15 an hour who come from socially disadvantaged backgrounds. But in return to that $15 an hour stipend, we developed a partnership with Miami-Dade County's foster care system. And these undergraduates are hooked up to these high schoolers in foster system on a one-to-one basis. So we sex match them, boys with boys, girls with girls. And these are foster kids who have very bad life outcomes. And like, guess how many go to college as a percentage of foster kids? I mean, these are kids who bumped from home to home, and they just don't want to be told what to do, especially college awareness. And so 4% is the national violence statistic for them. Like, they're just waiting to turn 18 and get out from under the man, right? My undergraduates, we had to do mentor training for my undergraduates to then make sure they're able to talk to these high schoolers who closed off. And we had 12 in the first batch. All 12 are in college now. One has dropped out. So 11 out of 12 are in college now, which was great. We have done this successfully for 2 years, and this year we got an anonymous gift from a donor. The gift was big enough to do this in perpetuity and open it up to undergraduates from FAMU FSU. So I'd be willing, if you guys were interested, if any of you, especially at the undergraduate cohort who's not here, if you'd be interested in participating in these outreach activities to foster youth in Miami. We had to do a lot of work, like a lot of approvals and a lot of work to make sure these at-risk youth can come on campus, come to labs, So we have that now set up through the lawsuit. And it's called the First Star Academy. The name of the program is called Joint Academic Nurtureship for Underrepresented Student Genius. And we will now, through this gift, have a lot of those students coming on campus this summer. And undergraduates from UN, but also the 4th largest HBCU in the country, which is FIU. Thank you.
It's a good way for our graduates to get involved with something.
Yeah, that's true. I need to find that.
Yeah. Cool. All right, let's thank Dr. Agarwal again. We have a